Hello, my name's Tom Jovic. I'm a plastic surgery registrar in the South Wales area, and today I'm presenting my research, which I undertook during a RCS Freemasons Fellowship about 3D printing. So I'll just quickly have an overview about myself, about the project that I undertook and the future directions my research has taken since doing the fellowship. So I undertook my RCS Freemasons Fellowship back in 2017 to 18, and the research title at the time was The Chondrogenic Potential of a Novel Nanocellulose Biomaterial, which hopefully will make a little bit more sense by the end of this presentation. So a little bit about myself. So I was at medical school between 2008 and 2014 at Cambridge University, and it was here that my interest in research was sparked during an integrated degree in which I undertook some laboratory research. Thereafter, I went to Edinburgh to do an academic foundation programme and have since come to Wales as a core surgical trainee and now plastic surgery registrar. And I'm currently in the middle of a PhD looking at 3D printing cartilage, which is a continuation of the work presented here today. Wales is a stunning place, and we're also very fortunate in that we are host to several high quality research institutions, including the Institute for Life Sciences based at Swansea University, and also the European Centre for Nano Health as well. I'm lucky to be part of a large plastic surgery research group known as the Reconstructive Surgery and Regenerative Medicine Research Group, which is based within Swansea and affiliated with the Welsh Centre for Burns and Plastic Surgery, which has a very large catchment area covering most of Wales and some of South West England as well. I was very fortunate to have good supervision during my RCS Freemasons Fellowship in the form of Professor Ian Whitaker, who is the head of the research group Recon Regen, but also from some of the biological supervisors, Professors Thornton and Doak, who are based within Swansea University Institute of Life Sciences, and also a couple of engineers, Professor Hawkins and Dr. Zhao, who were instrumental in getting this work done. So my title, as I mentioned earlier, was The Chondrogenic Potential of a Novel Nanocellulose Biomaterial. But essentially what that means is that I was trying to look at how well a natural ink allowed us to 3D print cartilage for facial reconstructive surgery. So the problem that we were trying to address is that 1 in 100 individuals are affected by disorders that affect the form and function of the face. And this can arise due to congenital reasons in the aftermath of trauma, burns, or malignancy. Now, one potential solution for these patients could be to have some 3D printed tissue to restore form and function. And we do have the technology to 3D print things that resemble an ear and nose in plastics, but also we have the potential to be able to isolate and grow patients' own cellular material in a laboratory environment. What we need to do now is combine these two things, and we need a good ink to be able to do that.
How are you doing?
So a bio-ink is a type of water-based gel that enables extrusion through a printer, but also cellular material to grow within it. And the problem with current bio-inks is that often they are rejected by the immune system, particularly if they're made of plastic, or they don't have that biological activity that allows cells to grow in the way that we would want. What we focused on was a material called nanocellulose, which is derived from tree pulp. And this is known to be well tolerated by the immune system, and also as a natural material felt to be better for growing time. So how we undertook this research was we were given bits of cartilage left over from nasal surgery, and we were able to isolate the cartilage cells from these samples and grow them in the laboratory, so that they could then be put in a type of biological ink, which was either nanocellulose or alginate, which is a commonly used seaweed derivative used in 3D printing research. And what we actually found was that the cells that were grown in the nanocellulose material formed cartilage almost 150 times greater than that of alginate when looked at the gene expression level. So it holds tremendous promise for cartilage tissue engineering. And this was also evident when we looked under the microscope as well. You can see on the left column there what native human cartilage looks like, and this is more closely replicated by the cells that were grown in the nanocellulose material. I was very fortunate to be involved in a number of opportunities during my RCS Freemasons Fellowship, including the writing of a book chapter and engagement in a clinical trial, the opportunity to present this research nationally and internationally on a number of occasions, and to contribute to the scientific literature in the form of research publications. And since the RCS Fellowship, I've been able to progress this research into my PhD, looking at trying to strengthen this material. So in doing so, I've combined the nanocellulose discussed earlier with something called hyaluronic acid, which you may have heard of. It's a commonly used injectable filler in cosmetic surgery, but also a native component of human cartilage. So the hope is that this would strengthen the material and make it even better at forming cartilage. And that is indeed what we've seen so far. And quite promisingly as well, we've been able to print this material in the shape and form of something that resembles a human ear, meaning that this has tremendous promise for patients in the future. What I'm currently up to now, so I'm in the final year of my PhD looking at cartilage tissue engineering. I've been very fortunate in being able to acquire some further funding for this research from Action Medical Research, Microtia UK and the British Association of Plastic Surgeons on the background of having completed this RCS fellowship, and further contributed to the research literature in the form of publications, presentations, and a textbook which is in progress. But more importantly, the impact this has for patients is tremendous. So currently we have to take out rib cartilage to be able to reconstruct these defects, such as missing ears and noses. And this is a lengthy, complicated procedure with a number of potential complications, such as damage to the lung and deformity of the chest wall. But the results can be very good. If we were able, however, to 3D print tissue for them instead, we'd be able to bypass this plethora of complications and give them an exact replica of their missing structure with their own cellular material, which would just be transformational for patients. Furthermore, this will provide a springboard to look into developing other tissue types through 3D printing, which include things like skin, bone, limbs, and even organs. So the next step, after completing my laboratory studies, I'm hoping to go to Harvard University to undertake some animal work, just to verify how well the material endures in a living organism. And if we're successful here, we'd hope to be able to progress this research into first-in-man clinical trials. Thank you very much for listening, and thank you for all of your support.